Everybody can hear me okay? Okay, good evening and yasu, everyone. It's an honor to be here. I want to thank Yanis and Helen, our gracious hosts tonight, for having me here and organizing this event, kind of last minute, I guess. Also, Marissa, my very dear friend who connected us when I told her I was coming to Cyprus. I also want to thank Daphne Christou, who is here tonight as well and is also hosting me in Cyprus. My name is Dr. WhiteFeather Hunter. I'm a Canadian artist and a biosciences researcher specializing in this radical intersection of art science and feminism. And I'm a badly behaved practitioner in biotech. So I want to share with you tonight how biotechnologies can be badly behaved or how we can be badly behaved through them by flipping the script on traditional science and cultural narratives and embracing feminist subversions in the lab. Am I doing like a, okay, maybe I'll stand back a bit. So I've been fortunate in my practice to work across many different disciplines. This is something that I like to call intellectual promiscuity. I hold a PhD in biological art. This was a cross-enrollment between the UWA School of Design and the School of Human Sciences. So I was working in both schools equally. So through this, I've been able to collaborate with leading biolabs and scientists worldwide to extend my art practice to a laboratory practice, or what I like to call laboratory craft. And you'll see an example of that in a moment. So this has been about 12 years of developing technical skills in bioengineering and microbiology. And my work is specifically meant to challenge biocultural norms. It incorporates feminist theory to sort of critique how bodies are used or how they're excluded in science and why. And so I'm challenging how traditional scientific research often excludes certain bodies and perspectives, especially those of women and other marginalized groups. So in bioscience, there's a tendency to create a sterile, detached environment. And this perpetuates the notion that knowledge production has to be sanitized and removed from the body. And so I reject this with my work. So I'm rejecting this, these sanitized narratives, by incorporating embodied, feminist, craft-based methods and very personal body materials, as you will see. So why do we need badly behaved biotech? Other than the fact that women's bodies and health are grossly under-researched in biomedicine and in tech development, I want to define what I am calling tech washing. So tech washing, like whitewashing or greenwashing, tech washing is a term for when new technologies are marketed using labels like eco-friendly or cruelty-free without addressing the deeper ethical and power issues behind the tech. So instead, we're presented with a one-dimensional solution to solve some world problem, without looking at what is behind that development. So tech washing is this hype. It's used to market technologies in really glorified ways. And a good example of this, and relevant to the work that I do, is the concept of lab-grown meat. Who's heard of lab-grown meat? Yeah? It's also called clean meat. So clean meat sounds like a really good moral solution. It's presented as an alternative to exploitative industrial farming practices, but it often hides the exploitative practices that are used to produce clean meat that's grown in the lab. So to just explain this a little bit, for those of you who don't know anything about how clean meat might be produced, the image that you see on the left is a bottle of fetal calf serum. This is a blood product. It's extracted from the heart of a fetal calf while it's still in its mother. A needle is inserted into its heart and it's essentially bled dry. And then both mother and calf are sent off to the slaughterhouse. So that blood, considered to be a pure, uncontaminated blood, is used to produce this blood product. And that is what's used to grow cells and tissues in the lab, and that's how meat is grown in the lab. It's essentially animal cells that are fed this nutrient serum until they become a tissue mass, and that's what humans are supposed to then consume as a solution to the world problem. So what I'm offering as an alternative, this is something that I was playing around with as part of my PhD research, and what you see is in the hand that's kind of coming out from that serum bottle is a tube of human menstrual blood serum. So I was investigating this because menstrual blood is a source of thousands of proteins that you will not find in any venous blood. It's really and truly sustainable and eco-friendly and cruelty-free. So that was my sort of taking a piss out of the biotech industry, and I'll talk more about that project as well. But essentially, I want to show you how feminist art can challenge these narratives, these sanitized narratives, by critiquing the storytelling, the hype of tech washing, and imagining a more inclusive or ethical approach to biotechnology. This is where feminist art is badly behaved in terms of disrupting, capitalist, tech washing, and my processes are also redefining whose knowledge counts in scientific innovation. So more on this to come. So before I get into those projects, I want to talk a bit about my background, how my practice is blending feminist theory and craft and biotechnology. So in my earlier work, before my doctoral research, which you see here, craft traditions like textiles, which are often associated with women's work, became tools for me to question and disrupt male-dominated structures in science. So this video is displaying a novel technique that I developed called wet weaving. This is demonstrating how I'm using woven structure to construct a scaffolding for tissue engineering. So all of the cells and tissues need something to grow on. So this is almost like a miniature architecture, which could feasibly be implanted in the body once the tissue is grown on it. So the plain weave structure that I'm using here in combination with the material and the tool, and I'm using a single horsehair from the tail of a Mongolian stallion. This is a discarded material used to make violin bows, so I was repurposing that material to do this weaving. It's a protein fiber. Cells really like it. It's being woven on a 3D printed microloom, and these together result in this tensile environment that resists droplets, and it allows for a slow capillary action. So just to explain that further, because that's sort of technical, when I'm later seeding these weavings with tens of thousands of cells, I begin by placing a drop of cell suspension into the center of the weaving. And cell suspension is essentially a bunch of free-floating cells in a nutrient serum. So I'm putting a single drop onto the weaving, placing the whole thing in a petri dish and putting it in an incubator for an hour or two, and during this time the cells start to adhere to the horsehair. Now, attaching to the fibers or attaching to the scaffold is really important for tissue engineering. And as these cells start to multiply, I noticed that they seem to mimic the structure of the weaving in the direction that they were growing. So cells, they have an innate tendency for self-organization. But I noticed that they were influenced by the work that I had done to construct them a scaffold. And so this showed me that they were responsive to their environment, but this is what these cell types do in our bodies. They're connective tissue cells. So they form a tissue casing around our muscles and around our bones to hold everything together, and this is also what they did on the weaving. And I'll show you that in a moment. So what you're seeing here, and a strategy of mine, is to present a lot of my lab work through video performance. It's not just didactic, it's also meant to display the labor that's required to conduct this kind of research. And both science and craft, in my case, laboratory craft, they're very laborious processes. This is about an 8-hour process in 3 minutes. So I'm also interested in opening up this conversation about the labor of the cells themselves. What are they doing? What are their efforts? What's what's involved in them having a sense of agency as microorganisms. So this is a concept that I come back to over and over again in my work. So what you're looking at here is a CO2 incubator. I built this from scratch using an old styrofoam cooler. It's got a magnifying window that visitors could look into and see the biotextile inside. The biotextile is within a bioreactor that I built. It's connected to 2 peristaltic pumps outside of the incubator. And so this feeds the nutrient fluid into and out of the bioreactor, and it works kind of like a digestive system. The biotextile inside the incubator is woven with strands of my own hair. And again, this was a protein substance that the cells really love, and it was easy for them to attach to and grow on. So this image shows the hair, and it shows how the cells are starting to form tissue on the weaving. They attach to the interstices of the fibers. They begin to grow in a spiral formation until they fill in the negative spaces of the weaving. So this is what it looks like when they fill in the gaps. So you could say I'm filling in a gap in scientific research here. So now I'll introduce some of my doctoral research, beginning with this project that I call Moon Calf Unclean Meat. So for this project, I used my menstrual serum that I showed you already, as well as stem cells that are also found in menstrual fluid. Now, these stem cells are a very special, very under-researched stem cell type. They are now being studied as a cure for Alzheimer's disease. They're also being used to treat damaged heart tissue after cardiac arrest, but they're still extremely under-researched because of the taboo that is associated with menstrual fluid. So I started researching with these because they were accessible to me, free source of stem cells. So I'm using my serum and cells to experiment with what I'm calling unclean meat, and this is to challenge the sanitized ideals that are presented with lab-grown foods. And it also questions who defines, cleanliness and purity. So by calling it unclean, of course, I'm confronting the stigma around menstrual blood while also critiquing the moralized biotech narratives. And this project has sparked all kinds of intense reactions at the institutional level, as you can imagine, showcasing how women's bodies are used or they're rejected in scientific innovation. I had to push through a lot of rejection for this work. So I'll tell you more about that later. But to give you a better idea of the work, I have a 2-minute video that I'll play. This has some audio, so I get a chance to shut up for a minute and drink some water, and then I'll continue talking when it's done. Okay, so as you saw, that research was actually the profile that was featured by a biotech giant, Merck, or Sigma Aldrich, for International Day of Women and Girls in Science in 2021, which was so funny to me because it was an art project. Anyway, you'll have noticed also the mention of witchcraft, and that's because this project is within my larger thesis research that's called The Witch in the Lab Coat. And witchcraft is a term that I'm using to describe deviant feminist behavior, especially regarding reproductive technologies in health, because women's bodies have been demonized for a very long time, especially when menstruating. And so my work is also a play on these old archetypes. The witch represents someone who works outside of institutional norms, embracing intuition, body knowledge, and taboo materials. So this metaphor is a really powerful tool that I've found for rethinking the lab environment and its power structures. Now what you see is the work presented here in 2023 at Art Laboratory Berlin. Aside from tissue engineering, I like to also build all of my own electronics and lab equipment that's needed to keep the cells alive outside of the laboratory setting and in a gallery setting. So you saw with the biotextile that I built my own incubator, which kept the tissues alive for a period of 6 weeks in the gallery. What you see here also has cells. There are menstrual stem cells growing in each one of these chambers. They're interconnected. So this is a bioreactor system. I worked with a glassblower who produced these vessels for me. This kept the cells alive for about 5 weeks before it got contaminated. So I like to say it got a yeast infection and I had to shut it down. So this gives you a better idea of the installation, and my lab coat was heavily worn, as you can see. So if you want to know more about that project, there's a couple of different papers, journal articles that are published on it. If you want to know more about what I discovered in my experiments, because there's much more than I can go into here, how it relates to the tissue and organ trade, as well as to witchcraft, I'm happy to give you the links to those journal articles afterwards, and it even includes my protocols, so you can try it at home. Now, before I go on to my next project, I want to show you a little bit of what I call tech porn. This is what scientists consider to be very sexy, high-tech imaging that's created of bioengineering experiments through high-end microscopy. So before I show you some of that microscopy, though, I want to explain that part of my research creation process is staging my experiments. I'm not just working on them in the lab, I'm actually setting up an aesthetic environment. And I do this through photography and through performance before I go on to do months and months and months of lab work. And so this is how I represent my work in a symbolic way first. And this is a way for me to think through what I want the experiments to accomplish. So the image that you see on the left is a photograph that's doing just that. It's showing a menstrual cup. This is how I collect the fluid and extract the stem cells. And in this image, it's become part of the apparatus that's used to grow them. The image on the right shows 3D bioprinted versions of the menstrual cup. This is printed with a gelatin alginate bio ink that I made. It contains the menstrual stem cells, and so this form eventually becomes tissue. The cells will grow and they'll consume the gel, and the result will be a menstrual cup that's actually made of human tissue. So this is what the bioprinter looks like when it's printing a form. Some of the gels are cured by UV light, so that's what the lighting is. This is a syringe that goes into the bioprinter. It's full of gel and cells. The bioprinter warms up the gel with the cells, and it extrudes it to print the forms. Now this is the sexy microscopy. This is the bottom lip of the cup, and every stem cell has been stained with a fluorescent dye. So the green shows you the cells that are alive. So every green dot is a living stem cell, and then the red dots are the dead cells. Now if you zoom in closer, you can see the cells on the bumpy surface of the printed cup. And so you can see that there are a lot of live cells. And then if we zoom in even closer, these are clusters of stem cells that are inside the gel. And so every single cell nucleus is stained with a fluorescent blue dye. So everywhere you see a blue dot is a cell nucleus, the center of a cell. And then we can zoom in even closer so you can see those cell nuclei a little bit better. So these images are from my initial bioprinting experiments with menstrual stem cells. And I want to mention here that this research was under extreme scrutiny. I was working in a mother and child research hospital, which you would think would be quite familiar with the substance. However, they forced me to sign a contract agreeing to never publicly name the research institute, never publicly name any of my collaborators because they thought it was a PR nightmare. So I've experienced similar obstacles at the University of Western Australia. This has delayed my research by months, but this is why I say I'm badly behaved as a feminist pushing this kind of work forward. Now we'll talk about Sentient Clit, the Pussification of Biotech. So this work is exploring the idea of synthetic sentience by 3D bioprinting clitoris forms that have been embedded with menstrual stem cells, but I've gone a step further in this case. I've transformed the stem cells into neuronal types, so brain cells, using specific biochemicals called growth factors, and I'll explain this a bit more. But first, this project is meant to ask, can synthetic entities experience intelligence or pleasure? Is intelligence part of pleasure? And can we replicate that in a lab? So, I'm pondering, in a lab-grown organ such as a clitoris, is it the same as lab-grown meat? So I'm opening up some ethical discussions about designing lab-grown entities that might be capable of not only responding, but also conscious pleasure. This is raising questions about autonomy, consent, and the purpose of research and the purpose of biotech advance. So again, as with the biotextile work that I did, I'm thinking about the agency of microorganisms, their complex forms, possibly capable of expressing themselves in ways that we might not fully understand, but perhaps we can get closer to understanding. So I've got two videos I'm going to show you. This project, again, involved cutting-edge bioprinting techniques. I was working with a $500,000 bioprinter, collaborating with some leading scientists and bioengineers. I was also teaching them my techniques as well. And the aim with the Sentient Clit was to blur the lines between the organic and the synthetic, human and machine, and punking reproductive material as a source of pleasure in the lab. So we need a new mode of technophilia.
Right, so how about we grow a clitoris from the stem cell out of our own menstruation gland? Well, here we are. This is our lab-grown clitoris from our menstruation blood. Question time. First question, is it a sentient being? Wow, what is a sentient being? It's the ability to feel things. Can you feel? If I touch you, can you feel anything? Can you feel if you're just a bunch of cells made of neurons and erectile cells? What do you think, Clito?
Well, the creation process is essentially the same, and ideally you want to put both in your mouth, so I guess you could say that.
Next question is, can it give consent? Well, consent usually implies willingness, but how can we know if it can't even speak? And how do we know if it's completely under my control? Thank you for watching. Please subscribe. For premium members, we provide a special service. We can share the recipe with you so you can grow your actual clitoris from your own menstruation blood. And what do you want us to do with this living clitoris? Write it down in the comment section and we'll make your wish come true.
So obviously we're spoofing OnlyFans there. For those of you who don't know, OnlyFans is an online porn channel, I guess, among other things, but that's kind of, yeah, so that's what we're playing with there. Next video so together with this video, the next video is a little bit more scientific. I'm actually in the lab doing some work, and together it'll give you a pretty good overview. But if you missed it, she was saying that female scientists recently discovered that snakes actually have 2 clitorises, whereas male scientists before thought that they were just scent glands and that animal sex was just coercive and didn't involve pleasure. So that just kind of shows you the potential gaps in research that exists. So, before I play the video, I'll say that the image that you see on the top right is a microscopic image of my first successful experiments with transforming my menstrual stem cells into a neuronal type called an astrocyte. And the forms on the bottom in the petri dishes are the clitorises that contain the menstrual-derived cells. So this will explain a bit more. You'll see what I do. Hi, I'm WhiteFeather Hunter. I'm an artist in residence at Cultivate and a visiting researcher at the Institute of Molecular Medicine. This is a partnership that has allowed me to bring my project into a laboratory context and advance some research that I had started last year. My project is called the Pussification of Biotech, and essentially I'm working with my own stem cells that I have extracted from my menstrual fluid. Because this is such an under-researched material, because it comes from a taboo body fluid, there's a lot of room for me to experiment and discover new things about how these cells work, and this allows me to negotiate some new creative outputs for my research. One of the things that I'm doing while I'm here at Cultivate is looking at how I can manipulate these cells through a process called cell differentiation. So I take the stem cells and I test them with different biochemicals called growth factors, and those growth factors will influence what type of cell these stem cells can become. So what I'm interested in the most right now is differentiating these stem cells into neuronal types. So the types of cells that are throughout our brains, there are many different types. as I've been here over the weeks, I've been testing these different growth factors with my cells to see how they respond, how they might change shape, how they might change behavior, motility, or the way that they move around in a petri dish. So I've had some interesting results. Not necessarily the results that I wanted to see, but that's also really important information. So what will happen with these cells once they become neuronal types is that they will then be put into a gel in a 3D bioprinter and printed into an organoid shape, specifically a clitoris. So the next cell type that I'm planning to differentiate the stem cells into are cardiomyocytes. Those are heart muscle cells. Each heart muscle cell will beat with a heartbeat together in a petri dish. So feasibly, I could co-culture the neurons with the cardiomyocytes in the clitoris forms to have a clitoris that twitches and thinks about it. All right, so I'm going to back up a bit now and tell you about another part of my doctoral research, and I'll end with this project. This was called Blood, Magic, and Biotech, and this is a project that I want to talk about because one of my collaborators is here tonight, Carlos Jabbour, who is a senior data scientist for the Government of Canada by day and a vaginal microbiome detective for artists by night. So again, I have a short video that I'll play to demonstrate the lab work for this project. Essentially, I'm sequencing the genome of my vaginal microbiome. It's got audio, but I'm going to talk while the video is playing, so I'm going to have to turn down the audio somehow. Oh, yeah, okay. All right. Can you hear me fine over that? Yeah? All right. So theories on the material composition of vaginal and uterine fluids have ranged throughout medical science's history. In the early 17th century, it was believed to be a rotten ferment in the uterus. Later on, it was believed to be a menotoxic cocktail of pathogenic bacteria. Now it's considered to be biohazardous waste in our current biotechnological era. But vaginal and uterine fluids, when they're absent of infection or overgrowth, are neither toxic nor hazardous, and they can actually be very beneficial. Some of the microbes that are found within the vaginal canal produce bactericidal and virucidal compounds, so they act as antibiotics and antivirals. And recent research has shown the benefits of vaginal microbiome transplant, so taking microbes from one donor and putting them in someone else to help cure up intractable vaginosis. However, this perception of menstrual fluid as a pollutant or dirty continues to persist. So with this project, I was looking at what I could possibly learn through this performance of self-quantification, essentially, looking at my own intimate micro-community of microbes. So I'm using biotechnologies here for self-empowerment through self-inquiry and sharing this knowledge. And this is because biotechnologies are really radically shifting the way we perceive bodies, all bodies. So women really have a stake at being in positions of self-determination in the biolab. So my sample collection was for 32 days. It covered one full menstrual cycle of 25 days, beginning with the onset of menstruation and concluding with the end of the following month's menstruation. This is the first time in vaginal microbiome research that anybody thought to do a full menstrual cycle. So it included ovulation phase, postcoital samples. These are considered to be really notable events that could impact the data. Hormonal fluctuations, introducing foreign bodies into the microbial ecosystem. So the methods that I was using didn't test for human DNA, only the DNA of the microbes that were present. I used a kit called DN-Easy Power Soil Pro Kit. And this is a kit that's designed for dirt microbe analysis, but it's also really useful for extracting RNA from other microbial niches, such as the genital tract. There were other samples that were being processed in the lab at the same time with my fellow researchers, and those were swabs from cadavers. There was one researcher looking at decomposition dynamics. There were also rape kit specimens that were being processed that were provided by the Quebec Provincial Police. So our samples were all mixed up, randomized, and coded and sent away together. And this was really interesting for me because there's a relationship between soil and cadavers and vaginas that's cultural and mythological. So, for example, the fragrance of hawthorn flowers. I don't know if you know hawthorn trees, but hawthorn flowers produce something, a chemical that's called trimethylamine. And so trimethylamine gives the flowers this odor that has been associated with the smell of sex. So in the spring, this flower is particularly associated with fertility because of the odor that it emits. Interestingly, there's also a bacteria in the vagina that also produces trimethylamine and this same odor, which attracts pollinators. So I found that super interesting. Part of the performance aspect of this work was looking at health performance. How does my health measure against health standards? And I discovered that RNA sequencing is not a perfect science. It remains really contestable, in fact. It's being promoted again, tech washing, as this frontier with tremendous promise, but it also has a lot of avenues for discrimination. So as I'm going to play this again while I'm talking. As I was watching Carlos sort of massage the data that I had received back from Genome Quebec, and understanding the rules that he was applying to the data, how he was shaping, the message essentially that was in the data, I was able to scrutinize things like biological determinism. We fall prey to this when we're looking at DNA results. It's often taken as fact about a person or a race or a gender even, and these kinds of biologism have led to suspicion of biotechnologies. They've been used to produce genomic proof of racist health hypotheses, for example, without any consideration of culture or socioeconomic realities or other things that impact the data. So here's a beautiful graph that Carlos produced. This shows you all of the species of microbes in my vagina. And to provide an example of what I mentioned about racist health stereotypes, these microbial ecosystems, the microbiome that's found on the skin, it's found in your gut, and in the vagina and other bodily locations, they really vary a lot between people. They vary a lot from day to day, in fact, which is why it's important to take samples over an extended period of time, not just one day. And they vary greatly between and within different socioeconomic groups and racial groups as well. So you can't just peg sort of one profile. What's considered normal for a vaginal microbiome can also shift a lot. So the main microbe in my swabs is lactobacillus. This is the same bacteria that's used to make yogurt. It's supposedly the normal one for white and Asian women. This is held up as the healthy standard, which is problematic. So where does this leave everybody else who's not white or Asian, right? However, the main strain of lactobacillus in my data is called Lactobacillus iners. And this is only a recently discovered microbe. It's not well understood at all. It's typically seen in samples from Black or Indigenous people. So its role in the vagina is not well understood. It could be harmful, it could be helpful. They don't know. It could be normal, it could be abnormal. Nobody knows. This is a lovely co-occurrence network. Again, thank you, Carlos. This shows where the microbes might grow together or where they might hinder each other. So they have a relationship. This shows all the main taxonomic groups of bacteria that were present in my samples. And Lactobacillus iners was fluctuating wildly throughout the month, and this matches what science knows about it so far. It's associated with instability. It's believed to colonize after the vaginal environment has been disturbed. So what does this indicate? What disturbance? Now, I'm repersonalizing this data. I'm making it about me because I know my own health history and there's valuable information to be gained from that. A disturbance that's occurred in my medical history is sexual assault. And this is the story with many, many women. However, it's even more so the case for women of color who have more socioeconomic challenges and it leaves them more vulnerable to forms of violence. So vaginal microbiome studies don't control for sexual assault. The question's never asked. It's a significant and it's an often hidden factor in many women's lives. Any data that comes from rape kits, like the ones that were processed with my samples, are not used in vaginal microbiome studies. They're only used for criminal investigation. So this is a huge knowledge gap in science. And so with my research and the performance of it, I'm repersonalizing this abstract data. This is a feminist act. I'm openly discussing taboo topics, such as sexual assault and menstruation, to highlight these research gaps and to destigmatize realities of women's health. So just to recap, this is the end. Some of the ethical and cultural implications. Who decides what's ethical in biotech? Whose bodies are prioritized? Whose bodies are being excluded? What information is being excluded? And how can art provoke deeper accountability in these systems? So here, the broader impact of feminist art is in challenging these dominant narratives, opening up space for new perspectives, fostering deeper cultural and ethical engagement with biotechnology. So the work's not just about critique, it's also about opening up possible outputs for these new technologies, opening doors to more inclusive, collaborative, and radical approaches to bioscience. The image that you see on the left is an AI prototype for a new 3D bioprinter I'm building from scratch in my kitchen back in Canada. So we're having fun with that right now. So yeah, feminist art such as mine is also activism. It's asking what happens when biotechnology isn't just driven by profit or patriarchal control, but by community empowerment. So I'll end there.